Hi and Assalamualaikum to Miss Akmayati. My name is Nurul Shahira binti Abdullah. My metric number is BPT 04230002. So for today's presentation, I will explain about lipid-based nanocarrier. So lipid-based nanocarrier is the nano-sized delivery system made of lipid, which is fat that can encapsulate the therapeutic agent, drug, protein, and DNA, and deliver them to the more effectively to the target tissue. Two of the most prominent lipid-based nanocarrier are the liposome and also the lipid nanoparticle. This is the difference between the liposome and solid lipid nanoparticle. For the structure, liposome are spherical vesicle with a lipid bilayer similar to the cell membrane, and they are composed of the phospholipid, for example, like phos- phosphatyl acetylcholine, which naturally form bilayer in aqueous environment. And for solid lipid nanoparticle, they are consist of the solid lipid core, typically made from the solid lipid like triglyceride or stearic acid, and the surfactant or stabilizer to prevent aggregation. They are typically smaller than the liposome, which is the 50 to 1000 nanometer. For the design, liposome can vary in size, which is 30 nanometer to several micrometer, which influence the drug loading capacity, drug release uptake, and the drug release rate, and also the cellular uptake. So the type of the liposome got two types, which is the first one is unilamellar liposome, which is the single bilayer, and they are also efficient in the delivering in a small molecule. And the second one is the multilamellar liposome. They are multiple bilayer and provide more stability and also drug loading potential. For design, solid lipid nanoparticle, they are particularly useful for lipophilic, which is fat soluble drug, and the solid lipid core is used to encapsulate the drug, ensuring stability and also potential from the environmental factor. So this is the difference between the unilamellar liposome and also multilamellar liposome. They are present in the many sizes and consist of one bilayer for unilamellar liposome, and also for multilamellar liposome, they are larger in size up to micrometer with two or more bilayer. So next is the type of solid lipid nanoparticle. Solid lipid nanoparticle with lipid cores. For the core composition, they are solid lipid only. For example, like triglyceride and also waxes. For nanostructure, they are mix of solid lipid and also the liquid lipid. For structure, with lipid cores, they are highly crystalline and also rigid. For nanostructure, they are amorphous disorder matrix with more fluidity. Next is about the drug loading capacity. With the lipid cores is more lower and limited to the crystalline structure, and for nanostructure they are higher due to the more flexible structure and also liquid lipid inclusion. Next is the release profile. With lipid core is more slower and more controlled release. For nanostructure is faster and more efficient release. Stability prone to crystallization and drug and exclusion over time. And for nanostructure, there are better stability, less prone and also polymorphic changes. With the lipid core for the application is the long active formulation, control release drug, and also can use for in the cosmetic formulation. For nanostructure lipid carries their high drug loading, gene delivery, and also vaccine. And the last is can use for dermatology also. So this is the mechanism of action for liposome. The first one is the drug encapsulation and release. Liposome can encapsulate both hydrophilic, water soluble, and also the hydrophobic, the fat soluble drug. The hydrophilic drug are trapped in the aqueous core, which hydrophobic drug and incorporated into the lipid bilayer. For the release mechanism, they are released from the liposome via several mechanism. For the first one is passive release. through diffusion depending on the lipid composition and the drug characteristics. The second one is fusion. Liposome can fuse with the cellular membrane, especially in the core of the lipophilic drug, and release the encapsulated drug directly inside the cell. So the last one is endocytosis. Cell can take up liposome through the endocytosis with a process where the cell enfolds the liposome and the drug released once inside the cell. So the last one is targeting surface modification. For example, like antibody or targeting peptide can direct liposome to specific cell, which is a cancer cell, that ensure the drug can delivery the right location. For the next one is the mechanism of action for solid lipid nanoparticle. The first one is drug encapsulated and release. Solid lipid nanoparticle encapsulate the drug within a solid lipid matrix. The drug is slowly released due to infusion from the lipid core, offering a controlled release profile, and the release of the often more sustained compared to other delivery system. For mechanism of action, there are two type, which is the Fikin diffusion. The drug move from the area of for high concentration inside the particle to the lower concentration outside the particle. The second one is matrix degradation. The solid lipid matrix may slowly degrade and leading to drug release over time. For the targeting, solid lipid nanoparticle be engineered to the target specific tissue by acting ligand to the surface. For example, improve specificity for the cancer cell. So this is the benefit of the lipid-based nanocarrier. First, can enhance the drug stability. Both liposome and solid lipid nanoparticle protect sensitive drug from the degradation caused by factors like light, temperature, and also the pH changes. The second one is can increase the bioavailability. So lipid-based nanocarrier can increase the solubility of poor water-soluble drug, making them more bioavailability when they are taken orally or inject. The third one is can reduce toxicity. By targeting drug to a specific tissue, lipid-based nanocarrier reduce the exposure of the healthy tissue to the drug and thereby reducing the side effect. And the last one is also control and the target release. Lipid-based system can be designed for sustained release, for example, like slow release drug over hours or day, and also which reduce the frequency of drug administration. Other than benefit, also have challenges of lipid-based nanocarrier. So for the first one, in also the production and scalability, manufacturing lipid nanocarrier consistently of a larger scale in a still is a challenge, and it requires precise control over size, drug loading, and also the stability. Stability issue. Sometimes liposome can lead to their content and also aggregate if not stabilized properly. And also solid lipid nanoparticle also can face issue with drug leaking over time, especially under specific storage condition. For the third one is also immunogenicity. Although lipid-based carrier are generally biocompatible, they are used in the body coat provoke the immune response, especially if they contain non biodegradable material. And the last one is also cost. The production of the high quality lipid nanoparticle, especially when the surface modification or complex design are involved, can be costly. So this is the real world application. In a cancer therapy, liposome formulation of chemotherapy drug such as doxil, liposol doxorubicin, have been widely used in the cancer treatment. This liposome reduce toxicity and healthy tissue and improve drug delivery to the cancer cell. For the second one is the vaccine delivery. Liposome are used in the some vaccine to enhance the immune system like liposome adjuvant. And the third one is gene therapy. Liposome, especially those with the cationic properties, are used to deliver nucleic acid, for example, like DNA, RNA for the gene therapy, and lipid nanoparticles are crucial for delivering mRNA vaccine like the Pfizer, Biotech, and Moderna COVID-19 vaccine. The next one is the topical drug delivery. Solid lipid nanoparticles are employed in the dermatology for the sustained release of the drug for the skin conditions such as lidocaine, for the pain relief or anti-inflammatory drug. And the last one is the oral drug delivery. For both liposome and also solid lipid nanoparticle can improve the oral bioavailability of poorly soluble drug like antifungal agent or antiretroviral. So this is the future trend in the field. Personalized medicine, nanocarrier for target therapy and advance in the molecular biology like enable of design nanocarrier can target specific biomarker and enabling personalized drug delivery strategy. For example, liposome can be engineered to the recognize and bind to the specific cancer antigen, delivering chemotherapy and precisely where needed. The second one is combination therapy, multi-drug delivery system, which is nanocarrier will increase use to deliver multiple drug simultaneously, which is such as combining chemotherapy with immune checkpoint inhibitor in the cancer treatment, thereby increasing the treatment efficiency. And the last one is biodegradable and biocompatible system for environmental friendly formulation as sustainability become more important. Researchers are focusing on the developing biodegradable lipid-based carrier and degrade safety of the body and do not cause long-term environmental harm. So for conclusion, lipid-based nanocarrier, which is liposome and also solid lipid nanoparticle, are at the forefront of nanomedicine, provide innovation solution to several pharmaceutical challenge. Their ability to enhance the drug solubility, improve bioavailability, and also reduce the effect, and enable target to the drug delivery make them invaluable in the therapeutic application. However, challenge in the light production scalability, stability, and also cost still need to be addressed. As nanotechnology evolve, future trends such as personalized medicine, smart delivery system, and uh, biodegradable carrier will revolutionize drug delivery and diagnosis. So this is all my references. Thank you.